All right, good evening. My name is Hunter, and this is Content Review for Principles of Biology students. Today we're going over animal evolution. So for this exercise, the best thing that you could really do if you're watching this in the future is look at trends as we go down these phyla and see what you can kind of pick up on. There's no reason to memorize this entire list, just make note of any emerging trends. So what can we learn about the evolution of the animal kingdom by examining modern invertebrates? Fill in the chart of the next 2 pages to organize the major characteristics of key invertebrate phyla, invertebrate not having a spine. Okay. So on our left column here we have characteristics, and on our top row, I guess, we have the key invertebrate phyla. Okay. So, first row, of course, we're doing examples of organisms in each phyla, and then we have number of tissue layers in embryo, tissue versus organ level development, true muscle cells, symmetry, cephalization, and then it goes on to coelom, type, digestive tract and type, circulatory system and type, nervous system and type, and then other. So we're going to start in each row and go across through all of the phyla and work our way through this. So a couple of these are pre-filled in for us to kind of give us a starting point. First we have porifera and porifera. The example here is sponges, and it looks exactly like what it sounds like. Cnidaria, C-N-I, Cnidaria, silent C. We have example organisms are hydra, corals, and jellyfish. Platyhelminthes, we have planaria, flukes, and tapeworms, which are a kind of flatworm. Nematoda, we have Ascaris, horsehair worms, these are roundworms, true roundworms. Annelida, we have earthworms, so segmented worms, leeches, fanworms, these are all segmented worms. Mollusca, these are all the really tasty ones, clams, oysters, snails, unless you're allergic to shellfish, my apologies. Arthropoda, insects, crabs, lobsters, and crayfish. These may be some of the most dominant invertebrates on the planet. And Echinodermata, we have starfish, sea cucumbers, sea urchins, and sand dollars. So number of tissue layers in the embryo. Embryonic development is the important distinction here because it, depending on which end forms first, that will change the sort of the destiny of individual tissues in terms of what organ systems they produce. So for porifera, doesn't apply. There are no tissue layers that we can distinguish between. Cnidaria, we have 2. We have an ectoderm outside and endoderm inside. So 2 distinct layers between, basically separating the organism from the outside world and the organism from its internal chamber. Platyhelminthes, we have 3 tissue layers: ectoderm, mesoderm, so ecto outside, meso middle, and endoderm. The mesoderm allows for the evolution of what will become circulatory systems and muscle tissues. I want to be careful how I word this, because while mesoderms give rise to circulatory and muscle systems in later organisms, that doesn't necessarily mean that we have those systems present in Platyhelminthes. So mesoderm makes possible the evolution of circulatory and muscle systems. We will come back to that. And then, I want to draw here. So those three layers, ectoderm, mesoderm, and endoderm, are the same for the rest of the chart through Nematoda, Annelida, Mollusca, Arthropoda, and Echinodermata. Embryonically, they all are made of these three distinct tissue layers. Am I going to get in trouble now if I want to type? There we go. So now we have tissue versus organ level development. Porifera, our sponges, have a quasi-tissue level. Cnidaria, we have true tissue level. And for Cnidaria, we have tissue level with the beginnings of organs in the reproductive system in particular. And then by the time we get to Nematoda, we have organ level. We have true organs. So distinct organs with specific functions in the body that we can really observe once we get to Nematoda. And remember, those are our roundworms. Okay. Trying to clean up the formatting, but I don't think it's going to work. All right, true muscle cells. Sponges, of course, have no muscle cells that are engaging in the kind of motility that we would traditionally think of. Cnidaria, likewise. But they do have epitheliomuscular cells that got broken up there, which is kind of annoying. Let me see if I can... Yeah. There we go. Epitheliomuscular cells. So cells that resemble musculature in the epithelium, in those surface level tissues. Platyhelminths. Yes, we have true muscle cells. And again, this is arising from that mesoderm. So while we have muscle cells through the rest of the phyla here, we have increasing levels of organization and complexity. So for Nematoda, we have longitudinally arranged muscles only. So longitudinally, all of them along the length of the organism. So if we're thinking about our roundworm, it has muscle cells, but they're all arranged in the same direction. And for Annelida, we have longitudinal and circular muscles. Circular muscles allow for peristaltic contractions, like you'll see later in digestive tracts. These might also be sphincters. Actually, I'm pretty sure that's correct. And then for Mollusca, we just have increasing complexity of muscle systems. So rather than just having longitudinal, you may also have this interweaving of muscle cells. Our cardiac muscles, intercalated muscle cells, are highly specialized to resist a lot of pressure, so they can maintain their shape. But that then remains true for our other two phyla. So talking about symmetry, and the two that you really need to remember is, does it have radial symmetry, where it's kind of the same no matter which side you look at it from? It may have a top and a bottom, versus bilateral symmetry, which is the one that you're all familiar with, because we are bilateral. If you create a plane of symmetry down the middle, we are more or less identical side to side. Sponges have no real symmetry, not even radial symmetry. Nor cephalization. So cephalization, cephalic, meaning head. So cephalization is the formation of a distinct head. So a sponge is just a blob of an organism with no real front or back or up or down. Cnidaria, so hydra, corals, jellyfish, these usually have radial symmetry, but no cephalization. And what's noteworthy about this is they all live a pretty sessile lifestyle. Sessile meaning they don't really move much, or if they do, it's drifting, planktonic we call it. So jellyfish don't have a lot of directed movement as part of their life cycle. They just kind of go wherever the ocean takes them and pick up nutrients as they go. So they're pretty opportunistic in that sense. Platyhelminths is where we start to see the rise of bilateral symmetry with concomitant appearance of cephalization and locomotion. So get out of here. With this bilateral symmetry and this formation of a distinct head, it is creating a body plan which is now specialized for directed movement, so this locomotion. So its life cycle can start to do things like hunt for food, which would be a distinct advantage over the other organisms around it, because if everything else is drifting and you can just go from one to the next, you become a pretty efficient heterotroph. Is there anything else I needed to touch on there? I don't think so. So this is the same chart, just continued. So a coelom is kind of going back to yes. A coelom is related to, but not quite the same as the tissue layers that we talked about with endoderm, mesoderm, ectoderm. So for Porifera, we have no coelom. Cnidaria, we have none. Platyhelminths, we have none. Nematoda, we have what looks like a coelom. It's not a true coelom because of the tissue that it's derived from embryonically. The gut is not lined with mesoderm embryonically. Annelida is getting even closer, but it's still not quite a true coelom. We have what's called a schizocoelom, formed by the splitting of mesoderm. And same for mollusca, same for arthropoda, and echinoderm. Echinodermata is when we see an enterocoelom formed by outpocketing of the gut. Digestive tracts. Do we have one? Kind of. Intracellular. Whoop. For sponges. So intracellular digestion only. You're going to have things like macrophages and other similar cells that are engulfing, maybe through phagocytosis, maybe through just the formation of vesicles, and pulling nutrients into the cell on a cell-by-cell basis. Cnidaria, we do have a vascular cavity, some extracellular digestion, and some intracellular. So gastrovascular cavity is just a cavity for digestion that's highly vascular, has a lot of circulatory tissue or structures around it so that nutrients can diffuse directly across those surfaces. So not super efficient, but more efficient than just relying on cell-by-cell digestion. Platyhelminthes, we have gastrovascular cavity, which is more highly branched, and the branching increases surface area, which increases contact with the foodstuff and with the circulatory system, so you're getting more efficient breakdown and digestion of nutrients. Nematoda, we have a one-way digestive tract. This is a new property. The gastrovascular cavities just kind of sit there. Food and waste and whatever else just kind of goes in and out the same way. A one-way digestive tract changes that. So complete from mouth to anus allows for more energy input per unit time. Can I change the font? Is it... I don't know that I can, to be honest. This is kind of an annoying thing about this particular software, where if I click from one box to the next, it's going to go back to whatever the default was. So in future streams, I will do my best to make it something more readable. Thank you for that feedback. Yeah, I don't want to dig through settings during the stream to change whatever my default is. Annelida. We're still looking at digestive tract. Yeah, stays the same throughout. Is the typing better or worse than my handwriting usually is? That's the real question. Because this is a lot of text to fill in small spaces. Moving on to circulatory systems. None for periphery. So everything is happening through diffusion. Cnidaria, we have mostly diffusion and some facilitation of circulation via fluid movement in that gastrovascular cavity. Whoa. Platyhelminths, we have diffusion, from some facilitation of circulation via fluid movement in gastrovascular cavity. And what's happening here? More of the same for Nematoda, but we have an addition here. Fluid movement is directed by a hydrostatic skeleton. So remember up here, Nematoda is where you had those longitudinally arranged muscles. That may have something to do with sort of the facilitated movement through that one-way digestive tract, which we talked about in the previous block. Annelida, we have a closed circulatory system. So starting to look a lot more like ours. So you have a heart or hearts, arteries, capillaries, and veins. So directed movement into and out of a heart throughout the organism. All right, so mollusca. Some species have an open circulatory system, unlike Annelida, and some have a closed circulatory system. For example, squid and octopus. So an exception to the trend here. All species of arthropoda have an open circulatory system. And Echinodermata have a closed circulatory system. So a closed circulatory system is not universally advantageous in the history of animal evolution, it looks like. There was some other factor that may have made an advantage for some ancestral lineages and not for others. Nervous systems. Sponges have none. Mostly B-cell communication. Cnidaria have what's called a nerve net. So, highly branched nervous tissue. Platyhelminths have a ladder-like nervous system with ganglion in the head end. Ganglion is a bundle of nerve cell bodies. Nematoda have lateral nerve cords. Lateral. Not literal. So lateral, if I'm facing this direction, lateral is side to side. And Annelida have increased complexity of the same. And that trend continues through the phyla. Empty box here for other. If there are other characteristics that you can see a similar trend in sort of increasing complexity, put those in here. This is kind of a prompt for you to go on a path of your own discovery here. All right, so using the information in the chart and in chapters 30 and 31 of your textbook, answer the following questions. What set of characteristics is shared by all of the invertebrate animal phyla in the chart? So what makes an animal an animal? All are multicellular and all are heterotrophs. So they heterotrophs, hetero, other, trophs is nutrients. So you are acquiring nutrients by consuming other organisms. Multicellular, of course, means they have many, many cells making up their bodies. What unique combination of characteristics defines each of the invertebrate phyla as separate from the other phyla? You want to go down each column and really do this exercise. Go through this chart that we just built and kind of figure out the defining characteristic of each phylum, making it distinct from the ones before or after. No single characteristic is going to define each phylum, and rather it's the specific combination of characteristics that is unique to and defines each phylum. The specific combination of characteristics that is unique to and defines each phylum. Yeah. If you compare the characteristics of one phylum of the invertebrates with the next, what key differences separate the groups from each other? Let's take an example. You'll need to compare the characteristics in a given column or phylum with those in the next. And those characteristics need to change from that box to the next. So, for example, annelids differ from nematodes in that annelids have both longitudinal and circular muscles and a schizocoelom, whereas nematodes have only longitudinal muscles and a pseudocoelom. In all other respects, at least those listed in the chart, the two phyla are very similar. Looking across the rows, what major trends appear to occur in the evolution of various organs or organ systems in these animal groups? Again, take some time with this chart. Really take note of those. And this chart is set up to allow you to visualize how the various systems changed in the evolution of the various phyla. And this arrangement should allow you to visualize major trends in the evolution as well. For For example, if you look at the number of body layers, it was a big one, really easy one. The number of body layers in the embryo, you can see a change from no apparent differentiation to 2 layers to 3 layers. Does this analysis provide evidence for or against the statement, evolution adds onto or modifies what already exists? And explain. It should be apparent from the chart. Modifications are built on what already exists. For example, we see that evolution from no clear separation in body layers to in the spongy embryo to 2 body layers in Cnidaria to 3 in platyhelminths. And the addition of the 3rd layer of the mesoderm allows for the evolution of muscle cells and the elaboration of the muscle systems through succeeding phyla. You could also look at cephalization and how that enabled more motility and also a greater need for more efficient digestion because you're taking in more and you're metabolizing more and you need to move more. Increasing complexity in terms of closed or open circulatory systems, one-way digestive tracts. All of those factors kind of allow for higher levels of specialization and complexity. And with that complex movement, you need more highly specialized nervous systems, right? So the chart organizes the major groups of animals based on grade or shared body plan features. What changes would you need to make in this organization to reflect the possible phylogenetic relationships uncovered using molecular evidence? So recent DNA analyses indicate that among the bilateria, so bilateral organisms, nematodes and arthropods should be grouped together. Recent DNA evidence analysis indicates that among bilateria, the nematodes and arthropods should be grouped together, and this phyla is called Ecdysozoa, and I would actually memorize this. Echinoderms and chordates should be listed among the Deuterostomia. Deuterostomia meaning the second hole formed first. Has to do with if the mouth opening forms first or the anus opening forms first embryonically. And the remaining phyla among the Lophotrochozoa. Figures 30.2 and 31.2 in your textbook will add some clarity to this. But I would memorize why ecdysozoa are important, that echinoderms and chordates should be listed among the Deuterostomia and know what Deuterostomia means, and the remaining phyla among the Lophotrochozoa. Know those. Numerous developmental characteristics are used to determine evolutionary relationships among animal phyla. What developmental evidence was used to link annelids, arthropods, and mollusks evolutionarily? All are protostomes. I believe this means that the mouth forms first. They all display spiral determinate cleavage. So, spiral determinate cleavage, and in the developing digestive tract, the blastopore becomes the mouth. And the coelom forms developmentally as a split in the mesoderm. What evidence is used to separate these phyla from the echinoderms and the chordates? Echinoderms chordates are deuterostomes. This means that developmentally they undergo radial indeterminate cleavage. Radial indeterminate cleavage. Yep. Mouth forms from a secondary opening opposite the blastopore. In the development of the digestive tract, the mouth forms from a secondary opening opposite the blastopore. So, deuterostome. So this is the way I remember it. Mouth forms second. And the coelom forms from the mesodermal outpocketing off of the primitive gut or archenteron. I have notifications beeping, but I don't know where. Okay. In biological terms, a group of organisms are said to be successful if represented by a large number of species or if the mass of all of the organisms in the group is very large. In both cases, large is determined relative to other groups or organisms. Given this definition of success, which of the major groups of animals would you argue is the most successful? And be sure to provide evidence for your argument. There are, of course, lots of potential answers to this question, and if you're watching this in the future, I would encourage you to take a second and think about this. Basically, pick some criteria and defend those criteria. For example, did you choose success based on the total mass of organisms, the total number of organisms, the total numbers of different species of organisms within a group? So like on one hand, because nematodes are found as ectoparasites and endoparasites on almost every living organism, they could be considered the most successful. Right? They show up in relationship with all other animals. On the other hand, if you look at the total number of known species, you might argue that arthropods are the most successful. There is a huge diversity in arthropods. Any reasonable argument is acceptable, as long as it is backed up with evidence that indicates why the group was chosen and should be considered most successful. So we kind of flew through that one. Last year, I think this one took me 45 minutes. So I'm going to stick around for a couple minutes if the folks hanging out have any questions. But otherwise, go get some sun on your eyeballs. It's a beautiful day. And I'll see the rest of you next week. Alrighty, take care. I'm going to let you go.